Hello and welcome to another Wolfram Institute live stream. Today we are joined with Christoph Steck, uh, presenting live from Germany at the University of Tübingen, where he's a medical informatics student. Uh, Today he'll be presenting his project from the Wolfram Summer Research Institute, um, and it will be on polymer folding. Christoph, take it away.
Okay, yeah, thanks for having me. You tell me if there are any issues, right?
We can see your screen.
Okay, perfect. Right, so I wrote up some code to work with what are generally called polymer or protein lattice models. And before we go into that, we'll take a short look at some protein structures so that we get a feeling for what's going on. So this is a really small protein. It's called ubiquitin. And on the top left here is kind of the full heavy atom resolution of the thing. So basically it's a embedded molecule graph, right? Then at the lower right-hand side, we have these more stylized representations, which are kind of highlight the more regular features that we find in these things. And these generally really come down to what are called generally called these sheets, where we have a parallel alignment of the backbone, and then we also have these helices, right? And these two representations in the middle here basically show the backbone only here on the bottom, and at the top we also see some side chains sticking out of there. And if you go here,
So,
we
so is this a secondary or tertiary structure for these proteins?
so this is the tertiary structure, right? So the secondary structure is a bit of a definition thing, right? You can argue about how real it is. Basically, more historically, one of the main regularities that have been found in these structures are basically the patterns of hydrogen bonding that you see, and these work really well for defining that stuff. And in general, you will find helices and sheets, right? So that's really what it comes down to, right? You can then, there in theory, there are pi helices which have a where each background residue binds to right, either 3, 4, or 5 residues further ahead. But in general, you will only find these alpha helices where it's +4 and then +5 or +3. You will find very rarely, right? So at the end of alpha helices and so on. And that's what you already can see in the structure here, right? You have helices and you have these kind of parallel alignments. Okay, then let's go on. Okay, so this is a smaller kind of summary thing. So this kind of protein, it has 76 residues, so it's really short in terms of protein, but still it has 1,200 atoms. About half of these are hydrogens. And if you look at the molecular graph, when it's not embedded in 3D, you really see that it's this kind of very long stretched thing, right?
So this long thing folds up to be the protein.
Exactly. Right, so this is another representation of what's generally going on with a lot of the kind of biological architecture. So there, so proteins are one of the more fundamental kind of polymers that you'll find in biology, and they are linear chains of amino acids. So at the bottom here we have lysine, which is one of the amino acids. They get chained up into these long chains, and then these chains fold up. Multiple chains can find themselves and kind of form long-term structures like this, and then these more long-term structures can arrange themselves into bigger units, right? So this here at the bottom is a viral capsid, right? And then these kind of connections, they are really not as well defined anymore, unfortunately, right? So organic molecules, they can really you can represent them really well as these graphs. They're really stable and so on, with these non-covalent interactions that do a lot of the work. That's not really possible anymore. So if you look at the kind of covalent connection graph of a whole cell, it will be super sparse, right? So it will mainly be small molecules, then small molecules chained up into larger polymers, which will mainly be proteins, DNA and RNA and so on. And that's basically it. And then most of the rest will be done by non-covalent interactions. And then you can have stuff like your cell wall where there are more covalent connections, but still they will be super sparse.
So just to be clear, can you go back one slide? Yeah. So these are individual proteins that are glommed together.
Yes, exactly. So basically, here you have three chains. Here you have three individual chains, and then these are basically the individual units that form bigger, in this case, whole surfaces and volumes, right? So the viral capsid in there will be DNA and so on, or RNA and so on, depending on the virus.
Mm-hmm.
Right.
Okay. Yeah, so that opens the door to different types of topological arrangements between proteins.
Exactly. Right. And then the general questions of the field are about the relationship between the sequence and the structure, in particular because we find that it's rather robust, right? So most of the proteins that we find in biology, they really robustly fold into a unique structure, but still that structure is very hard to predict, especially historically. Right now we are doing better with these AI models and so on, but it's still not fully solved, so to say, right? And then in particular when we get into dynamics and distributions, and these kind of main topics, they get particularly highlighted in the folding process itself, right? So if you have a final structure, you can assume it is rather stable. There are still dynamics going on and so on, but you assume if it folded there, then it will also stay there generally. But during the folding process, that's a lot more variable. And one of the questions would be how many unique pathways are there? Is it in general more like one pathway and so on? Or is it a lot of different things going on that converge into the same thing? Then going into the model, so we represent a kind of abstract polymer by a linear chain, right? So we right now we are ignoring the graph structure, the kind of smaller branches that would go out of it. And then we introduce conformational changes of that chain. So in this example here, between all of the vertices, we can change the kind of the bond angle between them. So each of these, from the straight arrangement, each of these things you can flip up and down, right? And then we get a kind of states graph. So there's another example, it's kind of bigger. Now we have two colors, and in the two color case, it's the kind of historical HP model where the yellow vertices are hydrophobic and the blue ones are polar. And also right now here we threw out the kind of double edges because you can always go backwards, right? And then taking a short look at the kind of hierarchy of the model resolution. So right now we are representing these polymers as linear chains. Right now they live in 2D. We have discrete angles and of course discrete time and so on, right? We have not really defined time in a sense, but there are discrete steps to get from one structure to the next. And then also, of course, we have very few vertex identities, right? And compared to that, the real thing, it's a graph, it's 3D, we have continuous torsional angles, atomic vertices, physical energy functions, so on. So this would be the kind of molecular mechanics resolution. And then additionally, you could define bond stretching and different bond angles, even though if you look at a structure itself, these will be very stable. The question is, of course, whether it's very relevant for the kind of vibrational modes that are going on while the thing is dynamic. Then to say some sentences about the model implementation, so the stuff that comes afterwards is easier to understand. So basically, you're working with internal coordinates. So each state, each conformation is represented by a single list. And then we use angle path to go from the internal coordinates to global coordinates together with an angle specification. And then to go from one state to the other, we use replace list, and this is fairly directly represented in the kind of code that I've defined. So a Polymer model consists of a sequence. These can also be multiple sequences later. And then together with a turn angle, which would be the 90-degree thing here, and state transition rules. And the state transition rules, they define what states are allowed. So these would be the entries in the internal coordinate list. And basically, there will then always be multiples of this angle at the top. And then we have three kinds of alphabets right now. This would be the uniform alphabet, which is not particularly interesting, of course. It's just useful to show the state space. Then we have the HP case, and then we have the split case where we split the polar into positive and negative.
So I get the listing out of how the chain is locally kinked. That was in the first like piece of code, and then you had a way to say like given a sequence of kinks in a chain, how to morph that into a neighboring configuration.
Exactly. So basically, you're just always doing all possible updates. But yes, exactly. And then the code is basically it's property based. So you define this thing. Internally, it's just an association, which basically carries specifications for the code, and then you use this kind of syntax to query for certain properties of the thing. So in this example, for this kind of Polymer model, these would be the minimum energy states, and they all have the same energy, right? So, and we see this same thing basically here. If you ask for the minimum energy, we get at the first position of the list that is returned, we get the minimum energy, so it's just -1 right now, and there are 4 states that have this energy. Then we can ask for the energy counts, so this thing has a super uninteresting energy surface, right? It's just 2 possible energy states, and I will talk later about how these energies are defined. Right, and then the most important property is the kind of states graph that we have already seen before.
So the energy is sort of how much it wants to stay or deviate from the configuration. I mean, you had like hydrophobic bits as the colors, let's say. So the colors don't want to get near each other, or they don't like being folded near each other sometimes, or as the case may be. So you're measuring that tension.
Exactly. So um, right. So basically, yeah, here's one. So in the most simplest energy function, the energy neighborhood, the interaction range is always 1. And in this case, with the 90 degrees, it's defined using a Manhattan distance. So this thing basically lives on a lattice, right? And then with an energy interaction neighborhood of just 1, we ignore sequence adjacent contributions, and then we just define a table of energies for pairwise interactions. So in this HP case, the only interaction term that is there basically is when two yellow things, two hydrophobic things are adjacent to each other, then they get a favorable interaction term of -1. So that's basically all that's going on there. Okay, then based on this, we can look at different angles and different state transition rules. So this is the 60-degree case, and the -1 and the 1 basically means that it cannot go straight. So the straight case would be having a 0 here too. And additionally, there are no sharp angles along the chain defined, so you cannot do 2 times 60 degrees, which would be a 120-degree bend along the chain somewhere. And then we can get these states graphs. We can make them a lot bigger. And for this 60-degree angle case, you already see some rather nice looking structures and so on. But then we can look in the 120-degree case. Here now also with the straight arrangements allowed. These are the same state transition rules that we saw with the 90-degree case earlier, only that it's 120 degrees now, right? And again, we see different kind of structures that are there, right? And now we also get these repetitive motifs, which kind of are like secondary structure, so to say.
Yeah, and then
And this is
how are the energies computed there? You don't have the Manhattan distance and the square grid anymore.
right. So we are still dealing. So the interaction range will still be 1. And with the 60-degree case and the 100, with the 60-degree case, which is kind of rather nice, the distance will always be 1. So you still get nice integer values, right?
Okay.
All of these vertices, they are always at a distance of 1. Right. And this basically gives us our most interesting case so far. And basically this is now the just the 120 degrees and the first 60-degree case that we saw before together, right? So it's 60 degrees and 120 degrees is basically 2 times 60 degrees, right? And then we basically get the same patterns that are possible with 60 degrees and 120 degrees and now mixed cases as well, right? So now into how the energies are actually defined. So these are the interaction terms that I'm using here. So we already had the hydrophobic case, and then the only other for the free color sequences, the only other favorable interaction terms is plus and minus, and then we get to unfavorable interaction terms, which would be plus plus and minus minus, right? And right, so here we have three structures. Do you see my mouse, by the way?
Yes.
Okay, so we have 3 of these structures that have an energy of -2. Most of them are, no, it's 4 actually. Most of them are still 0, and then there's a bunch of -1 cases. Then we can, of course, look at energy distributions. And this case here, it doesn't look at the the full energy distributions of a particular sequence, but it looks at the minimum energy distributions of a full class of sequences. So in this case, it would be all three color sequences of length 7, and then we delete duplicates by a forward and reverse symmetry, because in this model right now, where it's just a linear sequence and not a graph, for sequences that are the same forwards and backwards,
Yeah, they make the same.
sequences that are mirrored in that way, they are exactly the same in the model, right?
Mhm.
So in this case, we ask for the minimum energy for all of these. As I mentioned before, these return a list of the minimum energy together with the number of states that have that energy. And then we get a bunch of these, right? So one for each sequence that is in there. And then we can make this bubble plot of these tuples. Right, and it's not super obvious what's going on here, but basically there's a fair amount of structure here. There are -6 is the lowest energy that is appearing, right? Before we only saw energies of -1 and -2, but that also was a result of not running the states graph long enough, right? We only looked at kind of two stages. And at the bottom here are the energies that are unique. So whenever a tuple is appearing at the bottom here, then it means there's only one energy. The energy, the number of minimum energy states, the one in this case, right? And then there are some where basically all states have 0, even though the sequence is not that short anymore.
So this is a survey
Depending on you.
over a lot of different types of sequences.
Exactly. This is all sequences, all three color sequences of length 7. And again, we are now generally using the 60-degree angle case together with multiples of 2 also. So it's a 60-degree and 120-degree case mixed.
Mm. And there are no positive energies represented here.
No, it's only the minimum energy. For every sequence, we ask for the minimum energy. We get these tuples. The tuples are basically plotted here. The bubble size is how unique that tuple itself is. So if there's a big bubble, then a lot of sequences have that this combination of a minimum energy and a number of states that have that energy, right?
Can you compute somehow the roughness of the landscape, maybe just summing over all edges? And I guess if you maximize that, it means that it is very rough surface.
Right. We get to that now, basically. So yeah, right. So the next thing is to, all right, okay, so we look at some minimum energy states first, just how they look like. Right, so we saw before, these are not, they are generally not unique. Sometimes they are unique, and how many lowest energy states there are varies a lot, actually, right? So these are just some random sequences together with their minimum energy states, right? So in this case, there are 2 that have the same energy. Also 2, there's just 1. This is a unique lowest energy. In this case, there's a bunch of them, right? And also what is interesting, as they appear row-wise here, a lot of them are basically the same. So in this case, we see that they all have this same core here at the top, and then this kind of yellow-blue chain at the bottom is, yeah, it's dancing around because the yellow and the blue things don't have any favorable interactions, so there's no reason for it to stick on either side here, right? And then here we have another core, right? So we have different kind of core structures with more variable parts around them, and then there's really a lot actually for that structure, so very non-unique. And here's another case where it's less, but still a bunch. right? Then we can look at the energy distribution. So this is now on a per sequence base. The sequence is captured above, and then it's basically a histogram for over all possible over all the energies, right? So this thing here, it has the it's not defined anymore. Never mind. What's this? Right, so there's a property, it's called energy count. There's also all energies and so on, and this basically gives us what we want for a histogram. And then above these columns, there's one representative state plotted, right? And we looked at the kind of minimum energy states before. This is now all of them, right? So sometimes there's a unique minimum, as in these two cases, and then how many lowest energy states there are actually varies a lot, right? Then these are length 9 sequences. And also what we can now kind of see that is going on here is that generally the thing is trying to do phase separations, phase separation under the constraints that the kind of connectivities and the angles impose, right? So if you look at the thing here on the bottom top left, then with the lowest energy states, we see that the yellow things are kind of brought together as much as possible, and the red things are at maximum distance because they have unfavorable interaction terms. And then on the right hand side, it's exactly the reverse, right? This is how you get the highest energy. You bring all the red things together and you separate all the yellow things, right? And then we see, yeah, a bunch of interesting cases. And the kind of questions that arise here would be how much does this kind of energy landscape depend on the sequence lengths, right? So for very short sequences, we know the energy landscape can't be too interesting, right? Your maximum lowest energy will be rather high, right? It will be -1, it will be -2, something like that. And as the sequence goes to infinity, we generally expect that at least we expect that it gets stuck in the landscape, even though we have not looked at the landscape yet. So that's what we're going to do next. Right, then we can, of course, look at the state distributions. So how this works is it just takes the states graph that we had before, it takes the full states graph, so run for all steps. From this, we can take the adjacency matrix, right? And now we weigh each edge by a function over the energy distance. So we get our delta E term here, and we just say if the energy difference between the two states is 0, we assign 1. Then for smaller 0, we add the energy difference, the absolute of the energy difference. And if it's unfavorable, if it's an unfavorable transition, we do 1 over the energy difference basically, and then this is a row in our matrix, and we row-wise normalize the matrix, and we get this kind of Markov thing with our transition probability matrix that looks like this. So basically, on the left-hand side is our original state, it's our maximally unfolded state. And on the right-hand side, in general, as much state transitions as possible have been made. And indeed, we kind of see what we would expect, that there indeed is this kind of basin where the transition probability is kind of highest, right? So it wants to stay on that state. And
so
also
you're
the
saying
state,
that there are like local minima of favorable favorable configurations for a chain?
Right, so I have not looked at global minima. There often is, right, if you have a unique lowest energy, then you have a global minima, at least in the energy, and then it will usually reflect, will be reflected here also. And I have not yet looked at local minima. And in general, you expect that these will appear first. Yeah, there's a question of what's the first sequence where you find a local minima. In general, you expect that they start appearing as you increase sequence length, right?
So again, this is, you have a particular chain, and this has like 786 different ways that it can kink up and rearrange itself. And the initial state 1 is just the thing without any kinks in it, right?
Exactly.
And then you're measuring the energy. The color is saying, like, from this state to this state, it there's a single move that can transition from one to the next, and the energy, the relative energy difference is encoded in the values of this matrix.
Exactly.
Okay.
Exactly. It's a weighted adjacency matrix of the states graph. The number of rows or columns here is the number of states, so it's very explicit. And right, and then there's just a rather arbitrary weighting function, right? So it's not very physical. It's just, let's make it proportional to the energy difference, right? Right, and then we can take this transition probability matrix and apply it over and over again to an initial state to kind of get a state distributions over time, right? And then right, so then there are different kinds of plots that we can make to display that. At the top here is basically an array plot, so each row is the full state probability, state distribution probability vector, right? So this each row here has the same 786 entries. And we see this kind of sparse in here. And the plot below is basically the same. There are a bunch of different, basically it's a list plot with a bunch of different individual lists. And each list is the full state distribution vector at a certain time. So the x-axis, each point on the x-axis is a certain state. Again, we have 786. And this kind of peak that we get is over time what state. So there's one state, it's the same state that gets this kind of value here, the highest value here, which is kind of the energy basin. And it, after 1,500 steps, it accounts for about half of the probability overall states. And that's the general theme here. It's never 100%, which of course highly depends on these terms here, right, that you said. But in general, there will always be a distribution, but it's rather highly concentrated. Usually, depends on what does highly concentrated mean compared to the other states, right? But
That's
it
really
rarely goes
interesting.
over 70%.
So you
And
mentioned
then
it's like a
this,
Markov process earlier. So you have like an initial, you pick a state, a configuration, and then there are neighboring single defect configurations next to it that,
right,
and then you measure like which ones are energetically favorable, and you
right,
might have a probability saying, well, the best one will usually kind of make the fold, and then you see where that goes each time, and you'll get to these local minima, these basins that are sort of attractors in the state of in the state space.
exactly. It's just It's just a Markov process where the transition probability matrix is the adjacency matrix of our states graph, and we weigh it by some function over the energy difference between the states. Right. And so this is a nicer plot of the thing above, and each line in the plot is now a particular state, and the x-axis is now the time. So each kind of colored curve here is its own state and how its probability changes over time. And these are the probability of the 10 of the states that have the highest probability in the end. Okay, so there's really a very high concentration. We get this kind of nice asymptotic curve, and even though it's just 10 highest energy states or lowest energy states, half of them or more, they basically go to zero. And we have 3-4 states that dominate the whole kind of thing. Right.
Interesting.
Actually, it has to be 2 states, right? Otherwise, the probability wouldn't match up, right? So on the top here, it's 2 states, right? And then one of the more surprising things here is that it really takes a long time for these things to converge. So here we have, right? We can say about after 1,000 steps, it's basically converged. In this case, it takes a lot longer, right? So here we have 2,500 steps and so on. And again, we see the same thing. It's a different sequence now, and there's 2 states that dominate the landscape, right?
Wow, that's really cool.
Wait, so what are the energy values of those states? Sorry if I missed that.
Yeah, I should have put them there. I didn't. So I have not. Right, so looking at the energy histograms and so on, of course, would be nice. Yeah, I mean, I can do it now if you want, but I don't have them here.
Yeah, that's fine. We're just
Okay.
curious.
Yeah, no problem.
Right, I mean, that's one of the obvious things, right? So you don't have an energy minimum. You have multiple states that have the same energy. How does that affect the kind of landscape, right? And how much does the distance between those 2 states affect the energy landscape, right? If those 2 states are directly next to each other, that's a different thing than if there are 5 transitions between them, right? Because then the states that lie between them, they will get a higher probability, right, based on their neighboring states having a very low energy and high probability, right? So a state basically it gets some of the, if there's a low energy basin nearby, a state that is not itself low energy, it will feel the effects, right? So it will have a higher probability than you would otherwise expect. Right, and then onto the last part, basically. So this is, yeah, it's basically an interesting thing. You can define programs that directly operate on the structure of your thing. And the easiest way to set it up would be to take your sequence and your state and riffle them together, and then take these neighborhoods, right, linear neighborhoods in this case, and define an update sequence based on these neighborhoods. So you have rules of this kind where you say, well, if I find this sequence and this kind of state arrangement, transform it to this thing, right? And then you can apply that program to a sequence, and the red things here are states that are not allowed. and you get different kinds of states and so on, right? And of course, this kind of program here, it kind of lives in the wrong dimension, right? You would want it to be something that takes a two-dimensional neighborhood. But yeah, basically I thought that's an interesting thing, and I didn't work it out fully and so on. But basically the question to ask here is, are there programs defined in this way that define interesting sampling procedures that, for example, generally get us to the lowest energy state very rather fast, right? And then you can ask about the generalizability, how general is that program, well, it works on these sequences, but not on these sequences, and so on. And of course, the goal would be to find one that is very general, right?
So this is a pattern recognition. Looking for a subsequence
yeah, it's
in a particular
basically
configuration, then saying like the subsequence is gonna behave this way, or it can. So it's limiting the transitions in the state space.
right. So it's basically it's a 1D cellular automaton defined on neighborhoods in this kind of sequence, this kind of list. And this list now captures both the sequence of our polymer as well as the state that it's in, right? So basically it takes a neighborhood like this, and then it says go to the same thing, and it will generally, the way it's set up right here is that it will say change the middle value, right?
Mhm.
So take this kind of sequence and transform it into this sequence, and then the overall sequence, right, has an interpretation as this kind of our polymer model with a state, right? So it does straight state transformations on our thing. Okay, right then. Some interesting things we covered and ways to expand them, basically. So I think the 3D case is rather interesting and important, mainly because the correspondence is really clear, right? So with proteins, for example, it's really that helices and parallel alignments are the vast majority of regular structure, right? If you want to find other regular structure and so on, you need to look a lot harder, right? They are a lot harder to describe and so on. And also helices and sheets, parallel alignments of backbones and so on, they are very simple features. So you would expect that in the 3D case, you will be able to find them super quickly. And then, of course, there's still the general question with these kind of chemical polymers. How does their so that's a 3D case, and then graphs instead of linear sequences. And then once we are there, which is what I wanted to say before, is that then you can kind of define different polymer alphabets, right? So in the linear case, the whole state space and so on is kind of symmetric. If you're in the 3D case, if you get helices, left-handed and right-handed helices would be the same. Once you go into the into more branch graphs and so on, you can introduce a lot more asymmetry in the state space. And basically the kind of questions would be, can we find polymer alphabets that have very predictable behavior, right? So they basically always have one unique energy and so on. Then we can look into different angle combinations and how much they matter, right? We can look into the sequence structure relationship more generally than we did now. So for example, one thing would be, can can you define certain sequence motifs, right? So you just take a certain subsequence, you put it into different contexts, and you look at how often it forms the same lowest energy arrangement, right? Then in general, the kind of robustness. So one way to do that would be to just change the energy terms and look how much you need to change them to change what is defined as the lowest energy in your model under a certain sequence. Right, then we have not looked into this. It's also basically an obvious thing to do. Sequence similarity to structure similarity, right? If I mutate my sequence, how often do I end up at the same lowest energy structures, so and so on. Then this is basically the thing that the tiling with the folded structures would basically be the second stage of what we saw, or the third stage of what we saw with the viral capsids. So you define a sequence, you say this is the energy interaction terms and so on. Therefore, this is my lowest energy. Now that I have that and I assume it's stable, what happens if I put multiple of these next to each other, right?
Yeah, that's interesting.
Yeah, right.
I mean, in the case of like icosahedral viral capsids, like icosahedrons don't tile space, except that there are quasi-crystalline lattices which would conform to those angles. So that's
Exactly,
something
right.
to think about. It's interesting.
Right, so yeah, right. So one of the general biological things is, and then of course, if you have like your cell membrane case, which is a lot less defined, it's basically just it's a surface that closes in on itself, etc., etc. But with these proteins and these viral capsids and so on, you can have much more defined geometries. And in the membrane case, that will then be controlled by your cytoskeleton and so on, right? So it's still you can have very defined structures, but they are more flexible, right? They are not defined by the oily peptides in your not peptides, the things that your membrane molecules themselves, right? It's the cytoskeleton. But of course, with these proteins, you can have much more robust general structures and so on, which is an interesting thing. And then we can look at some other things which are potentially interesting. And one of them would be other ways to now work with actual real protein structures and kind of put them into discrete descriptions where we can do operations on and so on, which get us certain nice properties. For example, distributions and so on, they are very easier to define, right? You need some equivalence. What even is defined as the same structure and so on. If you're in a continuous coordinate space, then that's kind of a hard thing, right? And one of these would be just be nearest neighbors, right? So then you get these kinds of graphs. And so, for example, this graph looks a lot like the same graph that you would get if you have the red edges, which are nearest neighbors here, would be hydrogen bonds. So you see here, you already see the kind of the sheet-like structure here in the middle, and you see some helix-like structure and so on. And that would be a kind of discrete.
The red bonds would be so some of those are going to be like covalent bonds, and then other ones might be ionic bonds.
Exactly. So the blue edges here are so this is now at a backbone level resolution. So each amino acid gets one node, and so these are basically peptide bonds between different amino acids. So the blue things, they make a linear sequence, and the red things are nearest neighbors, as defined by the distances between the C alpha atoms.
Okay. And each node here represents an amino acid or an individual atom?
An amino acid. You can do the same on an atom level resolution, right?
Yeah.
But yeah, and it's potentially rather interesting to do it on an atom level resolution. And one of the things that we generally find, so this is a much simpler case, it's a very small protein, it has 20 amino acids or whatever, is that when we add these nearest neighbor edges under a spring embedding, the automatic coordinates that we get are very closely resembling to the actual coordinates. So these are, right, and this is very stupid kind of layout, just with a few extra edges added, right? So the coordinates here are the actual C alpha atom coordinates of the protein, and we see on the top here the thing is much more stretched out than on the bottom. On the bottom it's a helix, and we basically see the same thing in this graph that only has connectivity information, right? It does not inherit coordinates. And then one of the ways to set it up would be to initialize it, right? As this kind of graph where the red edges are just self edges, and then you update it continuously, and basically at each time step, the thing has to make a choice, and it's allowed to move the red edge one step further, right? So for example, it could take this kind of nearest neighbor red edge and move it to a sequence adjacent thing. And then at later stages, it could also jump over other nearest neighbors, right? So that's something you could do, right? And then you can do the updates, yeah, graph neural networks, message passing, and you can also probably rather well define this kind of simple programs that define a sampling procedure, basically, right? And then another thing which potentially could work, right? You could embed the molecular graph into a Wolfram model or another thing where basically the space moves, right? So you embed it in a kind of a regular graph. The molecular graph itself is constant; it doesn't change. The covalent bonds are assumed to stay the same, and the kind of the graph that defines the space around it can move and so on, right? And then another thing which we basically saw is that it's a special case of phase separation, right? Under the constraints that the kind of backbone imposes and so on. And there you can also do, yeah, other interesting things, right? Where these things come together and so on. Okay, that was it.
Yeah, that's fascinating. Yeah, it'd be interesting to look at three-dimensional configurations and see what kind of
Right.
dynamics, if they're the same sort of attractors and the Markov process there as well. I'm sure there
Right.
would be. I really like the analysis you did with the adjacency matrix, and kind of thinking about looking at the state space and then seeing how the attractors like really pull in from their neighbors. Be interesting to visualize that.
What's
Very
the
cool.
connection between the phase separation you were showing at the end and the protein models?
Right. I mean, yeah, it's not super strong actually. So what's going on here? This is just a 2D cellular automaton, so it doesn't have it doesn't conserve colors, but basically it's the majority voting one, right? So this one kind of preserves the number of edges and so on, the number of colors. Right. Basically, this is a way to set up a model of phase separation, right, in an unconstrained case. So each of these little cubes can move around. In this case, it doesn't even move around, right? The thing that is defined changes its color. So The way to make the relationship closer would be to have these little blobs that actually move around and not places in the grid that change their identity. And once you
I
have
see.
little
So you're saying a
blobs,
mass conserving rule almost.
right, exactly, right.
Okay. And then what?
Then you define interaction terms, and so one obvious way would be to make it probability based, right? So it's probably the standard way it's done and so on, right? These things have been studied before, I assume. Then you define a probability for sticking there, right? Because as soon as you have 100 times 100 grid, you cannot do all possible updates anymore and so on, right? And then basically you have the unconstrained case of what we have with the Polymer model above, where the Polymer model disallows for things to move away from each other, right?
Okay. Starting to get it. Thank you.
Right, right. And another way to look at it, if you put little blobs that are allowed to move around on a grid and have a certain probability of sticking together and so on, then you instantly think about, well, what about mixing them together? And then you get into the case, into the tiling case that we had before, right? So you have a polymer, as you say, under these energy assumptions and so on. This is the lowest energy structure. And now we get into this tiling case where we look into how we can arrange these together, and under the same energy assumptions and so on, do they stick together? And then that's basically the same case again, just with bigger things, right? But diffusion is kind of hard to implement. It's annoying.
Right. It'd be difficult to decide which pixel represents which configuration.
Right.
Unless you did a cellular automata like that on the states graph.
Right. I mean, so the polymer model works with internal coordinates, right? So there's no global space it's in, right? There's no global position. But as soon as you say, well, we now have two of these, then you need a kind of global coordinate space where they have relative orientations towards each other and so on. And in general, the kind of diffusion case, it's very easy to implement with these little blobs that just move around on their own grid. But as soon as you introduce blobs that consist of multiple other blobs and so on that are tied together, then it's not obvious how you do the diffusion of the bigger blobs in regards to the smaller blobs, right? Because in general, you would say, well, they have higher impulse, higher inertia, and so on. They cannot wander around as fast as the smaller ones, right? But you have discrete time, so you cannot say, well, the small blobs, they move an entire step, and the big ones move 1/10 of a step, right? Because then you get all kinds of issues. So that's in general why, for a lot of these things, graphs are in principle nice, right? Because you can change these edge connections freely. They have less interdependence between each other.
Brian, did you have any questions?
Yeah, first, this is great. About 6, 8 months ago, I tried doing a much dumber version of the same thing, and I didn't get anywhere near as far as you did. That if you scroll back to one of the beginnings of it, one of the earlier images? Yeah, like one, yeah, like one of those. The thing that I had done was the way that I was doing transitions was not based on the kind of thing that you did, but instead, um, I'd pick a node and I'd only let it move to a diagonal place if it didn't break the chain. Okay, so all of the moves were very local, and it didn't work that well. That if you go
Right,
back and if you go back and look at one of the square ones, it's more obvious with one of the squarer ones.
right,
Yeah,
right. So basically, you did
yeah.
the random
Basically,
walk
with these, yeah,
version.
the allowable transitions were only like if you look at the one here, if you move the mouse, not the top one, but the one down to the left of the top one. Um, Higher up, just the one second row. That one, I'd let the corner one move into the inner corner from the outer corner.
Okay. All
Okay,
right, right. I see.
right. Because the idea then was it felt like it was closer to physical reality. But like I said, I played with it, but I wasn't really getting anywhere.
Right,
The
right. I mean, one thing here that is kind of totally ignored is the kind of inertia case, right? So you say,
Yes,
well, these changes where you just basically switch one node um, relative to the rest has the same probabilities, the same
right.
properties as when you do a switch in the middle of the chain, right?
Yeah, right. And in fact, it could be that intuitively I wasn't doing the kind of switches like you have from the first row to the second row, because in an actual molecule that would be shifting a ton of stuff around quite far, quite fast.
Right, but I mean, in the end, you often do, right? If you look at these proteins,
Uh,
um, the
no,
middle, the middle portions, they still change their torsional angles, right? Even though maybe not at one step, and depending on the outer.
yeah, no, that was a minor comment. A more interesting thing that I thought may be interesting to discuss is if you when you look at protein structures, and you've obviously looked at a lot of them, there's kind of short range interactions and medium and long range interactions. Like the actual proteins like forming sheets and helices, right? And
Right,
it feels like the sheet helix formation is basically the choice of adjacent amino acids, that just set an angle. And, the decision to make a helix it's really going to be something very, very local. It's just you've got a consistent angle between pairs over a long distance. So it's kind of
right,
like, this is just an intuition that the tendencies to make sheets and helices is something that's very short range, but the overall structure is something much longer range.
right,
Well, Brian, for helices, it's definitely short range. It's on the order of about 4 residues. But for sheets, it's a series of long range interactions where you get the two strands aligning with each other, but they could be, dozens and dozens of residues away from each other.
It's true, but Bob, I wouldn't say that it's long range interactions as much as long range patterning. Okay,
Okay.
because yeah, because the sheet and helix, the sheet helix distinction really is from the sequence, you kind of can predict it, right? And it's whenever we're looking at the structures of actual molecules, it's like the actual activity, what the molecule is trying to do, is not built up out of amino acids, it's built out of sheets and helices.
Mm-hmm.
Right. So there's like a higher level kind of structure going on.
right. I
Yeah.
mean, one thing that is basically true is that proteins give you a different kind of energy landscapes than your standard polymer. So the thing to look for would be kind of in the general class of possible linear polymers of organic molecules that you can construct, which of those have fairly predictable behavior and strongly prefer certain arrangements. So in general, it will probably turn out that if you take more general linear organic polymers, a bunch of these will show far more interesting behavior
Well,
But
no, it's true because other than proteins, RNAs occasionally, if you buy into the RNA world, it's also getting into some of that, but nowhere near as elaborate. But it's kind of like you'd mentioned in a couple of points about the choice of an alphabet. And maybe the thing that I was saying was an intuition, is there's two layers of alphabets. There's the short range alphabet that is actually giving you something more alphabet-like for the longer range interactions.
yeah, I mean, I don't know. I don't exactly know the role between long and short range interactions, right?
Yeah,
So
it's because one of the things that's going on here, and for people who haven't actually spent much time looking at the kind of things that these are building up are cosmically huge. Right, that, Bob, you're kind of doing chemistry and the kinds of things that we've always talked about with what your work is. It's dozens to hundreds of atoms of a time where some of these enzymes are tens to even hundreds of thousands of atoms. And
yeah,
they managed to robustly do the right thing. So, by the way, I should ask this as a question, not make this as a comment. Um, the the capsid case is actually fairly rigid structure. Most of the actual protein cases are relatively floppy.
right. I mean, yeah, I think there's a fairly big range, right? Some of these make super rigid structures, and others are fairly floppy. Yes, and I mean, yeah, that's what's going on there. There's a big range in energies, even on the inner intramolecular scale, right? How stable is the protein, right? That's another way to test it, and so on. When does it unfold? When you heat it up, right? These values they have huge discrepancies, right?
Right, and that's not even beginning to talk about conformational changes, right?
Right.
Which is another layer on top of all of this. Now, again, I think that this is terrific work. The only comment that I was making is it feels like um, there are different distance scales that are important in different ways. And
Right.
especially for the bigger ones, it feels like it's a large collection of local things or semi-global things rather than things that are truly global. That the structures are big enough in question, are big enough that it's hard to imagine that the whole protein is optimizing for minimum energy rather than various corners of it independently doing it.
Right. I mean, it's not optimizing it just, but that's the main thing. Why does it predictably fold into the same unique structure, right? That's kind of the core question
Yeah,
in
and
general.
again,
And
I
why,
could
if
just
that's so, why is it so hard to predict, right?
Right. No, it's funny though. The only thing, and this is repeating what I was saying earlier, predicting that this string is going to form an alpha, is going to form a helix or a sheet, is probably a different kind of prediction than predicting that the whole thing is going to form a pocket.
Right. I mean, one is a bigger class, right? I
Yeah,
mean,
and they're very, very different scales.
Right. It's
So
basically between secondary and tertiary structure.
Yes.
yeah, I
Sorry, I
mean,
cut you off, Dugan.
yeah, so the way proteins form in biology is sort of you have this enzyme that's tacking on amino acids to the protein one at a time. And so it starts at the beginning and then it goes to the end of the sequence. And each time the protein folds to like the next minimum configuration. And that's usually um, predictable or it's repeatable every time the protein is made. Is that correct? That's my understanding.
I
No,
mean,
it's not. Number one, as you're adding new residues, there's not that much freedom of motion at that end. Because the penultimate residue and the one before that are still pretty close to the ribosome, and I believe that a lot of the folding is managed by what are called chaperones. That the secondary structure, helices and things like that, probably form pretty much on their own, but then the overall fold is often guided by these other proteins called chaperones. That that's what I last remember reading, but that could be a while ago now. Brian, you seem to be a little more up on the recent literature than I am.
Yeah, and it's again, I think what you it's sort of in responding to Dugan. It is, and maybe this is again what I'd been saying earlier. The secondary structure kind of self-assembles out of the ribosome, but the tertiary structure is it's a different order of mass. Sometimes, and I don't think I like whether or not the chaperone molecules appear or don't appear is, I think, fairly case specific, and it's not clear what kind of general rules you could make about them.
Yeah.
I mean, yeah, so the secondary structure, historically, you can probably use other more complicated geometric kind of descriptions, but historically it's just the hydrogen bonds between the backbone plus actually the disulfide bonds, right? So that's what's historically defined as the secondary structure. And then I'm rather sure that basically always when you have a full description of the secondary structure, meaning a full list of hydrogen bonds, you will find the tertiary structure. So there are cases of secondary structure where you only have disconnected helices or whatever, and you don't know the global fold. But in general, there will be some hydrogen bonds that define that relative arrangement rather well also.
Because actually, not meaning to overcomplicate. It says, Bob, you mentioned ribosomes. Ribosomes are another thing that manage to very reliably reproduce themselves very exactly. And they have a bunch of RNA mixed in with the proteins. And that seems to not hurt, maybe even help.
Yeah, right.
Yeah.
I mean, the ribosome is primarily at the core. It's a ribosome, right? It's RNA, and then the thing about the chaperones is that there might be rather sophisticated cases where these chaperones actually do a lot of work and they make the process a lot more reliable. But the main tendency that it's a self-organizing system is very, very strong, right? I mean, as soon as the space, that's probably one of the main things that the chaperones prevent. As soon as you have a very crowded space, you have a lot of proteins that are kind of in their folding process and so on, then you will tend to get a lot of agglomeration and so on. And I think that's probably the main thing that the chaperones do. And yeah, the process is remarkably self-organizing, right? I mean,
Mm-hmm.
Oh, very interesting stuff. Thanks once again, Christoph, for presenting, and
thanks.
it's a fascinating look into minimal models for protein folding. All right, gonna close down the stream. Thank you once again.
Thank you, Christoph.
Thank you.
Thanks, Christoph.
Thanks for